follicular dermal cells as potential contributors to advanced wound resolution.
Okay, thank you for the introduction. So I'll just get started and give you a bit of introduction about the project that I'm presenting. So as I'm sure you all know, the normal wound healing response is that you get an immune response, there's restoration of tissue integrity, and then both these processes resolve. And this normally results in a scar in adult mammals. Sorry. However, there are instances where you do see scarless healing and regeneration. So mammalian embryos, and that is often preserved in young children. For example, if they lose at the top of the digit in young children, it can regenerate if the wound's not closed surgically. And then there's model organisms like the salamander, which exhibit regeneration of non-critical tissues. However, in adult mammals, there are certain tissues that also display this scarless wound healing response. So the whisker follicle of mouse and rat both demonstrate this. So on the left here is a wounded follicle. So it was punctured with a needle into the dermal papilla, and you can see here that this dermal papilla is dead, and there's evidence of scarring developing up here. However, in a similar follicle that was left in situ for 21 days, the papilla has completely regenerated, and there's no evidence of scarring observed in the follicle. So I was interested in this effect, and I thought maybe one of the cells, or one of the ways this is coming about, is because of macrophages being associated with the follicle. So apologies if there's people who understand lots about macrophages, because this is a very, very basic description, but it's good for people who maybe aren't so sure. There's broadly two categories. The M1 type of macrophages are the classical post-defense macrophages that you probably know about, with an inflammatory response. And then there's an M2 grouping, which contribute to wound healing and regulation of the wound resolution. And that's nicely described in this paper here if you want to read more about it. But there's also another nice paper that really, really clearly demonstrates the importance of macrophages in wound resolution. So as I've said previously, salamanders are capable of regenerating non-vital tissues. So in this paper, they depleted the macrophage count in a salamander and then chopped off one of its limbs. After 40 days in the control salamander, the limb regenerated completely normally. But in the macrophage depleted salamander, there was no regeneration, and in fact there was fibrotic deposition in the wound site. And they took it a step further, which really, really nicely improved it. They allowed the macrophage count to regenerate, and then re-wounded on the same site. And in both instances, the limb regenerated completely. So it really, really nicely showed the importance of macrophages in wound healing. But obviously that's of no importance unless there's macrophages associated with the hair follicle. So these are some mice vibrissa follicles, and you can see down here, this is staining for F480, a pan macrophage marker. There's lots of macrophages associated with the dermal sheath, around the base of the dermal sheath, and even some in the papilla itself. When we wound the follicle, so this was done by chopping off the end bulb, the number of macrophages greatly increases and they actually invade into the hair follicle itself. So the aim of my project was to look at how macrophages contribute to scarless wound repair. We know they're abundant around the follicle, especially when wounded, and we think that they achieve this by the M2 macrophages secreting TGF-beta3. So what I'm presenting to you today is data looking at the human dermal cell contribution to wound healing. So we want to see if the dermal cells secrete TGF-beta3 in response to macrophage stimulation, to see if there's a sort of synergistic effect between the two cell populations. And then a lot of my other work is involved is looking at hair follicle induction, in which we use 3D cultures, which we believe are more similar to in vivo settings. So we also just wanted to compare and see if we saw a difference response between 2D and 3D cultures. So very briefly, we did that by differentiating a THP, the monocyte cell line, into M1 and M2 macrophages. We stimulated the media with LPS and then harvested this conditioned media, and then we treated 2D and 3D cultures of dermal cells with it and measured the secretion of TGF-beta3 via ELISA. So this first set of data is comparing 2D cultures of DS with 3D cultures, and these are, everything from this point is human cells, I should say. So what stands out is that the 2D cultures secreted significantly more TGF-beta3 than 3D cultures in all instances. But that's an artifact of the cell numbers used, so we use 10 times more cells in the 2D cultures than we do in the 3D models. By correcting for this, the significance is lost in the M2 stimulated category. In the 2D cultures, however, we didn't see any differentiation between the treatments at all. although this M2 population secretion level was elevated, it wasn't significantly so. However, in the 3D model system, we saw lots of significant differences between the treatments, which I will go into on the next slide. So we were happy using our 3D model, and we decided then to investigate the role of the response of the various different dermal cell populations. So these are DP cells. M1 stimulated, so a pro-inflammatory macrophage, induces a slight increase, but not significantly so, whereas M2 macrophages, the wound-healing macrophages, induce a significant secretion of TGF-beta3 from dermal papilla cells. This effect was seen in dermal sheath cells. The effect was actually even greater, so we saw a larger increase and a larger total amount. And there's intrinsically, dermal sheath also secrete more TGF-beta3. And then finally, looking at dermal fibroblasts, they secreted lower levels across the board, and there was no significant variation in the dream treatments, even though, again, this M2 stimulated category was upregulated. Comparing between the cell types, dermal sheath and dermal papilla control cells secreted significantly more TGF-beta3 than dermal fibroblasts alone, and the same was true when they were both stimulated with M2 media, so the alternatively, the M2 macrophages, the wound-healing ones. But one thing that was really interesting that we spotted in this data was that dermal sheath cells actually respond negatively to M1 macrophage conditioning media. So pro-inflammatory media actually made dermal sheath secrete less TGF-beta3, suggesting that they're very, very responsive to the types of macrophages surrounding them. So I'll just conclude and summarise and talk about maybe what further work we're going to look at. So the 3D culture system that we used displayed a significantly varied response, so we were really happy with that, as again, it's further proof that the model system is good and provides a more in vivo-like system. The dermal sheath and papilla, but not the dermal fibroblasts, could be potential contributors to scarless wound repair. The DS responded more strongly to M2 macrophages than dermal papilla and dermal fibroblasts, but on the flip side of that, they were also responding more negatively to M1. So we're not sure what that means yet. So in the future, hopefully what I'm planning to do is to develop a human model of hair follicle wounding, so I can observe the different types of macrophages in the follicle at different stages of wounding, and then hopefully see if there's any applications for this scarless wound healing that we see with dermal sheath and other systems. So one idea is maybe to apply them to non-healing ulcers topically and see if they can sort of help in that regard. I'd just like to finish by thanking everyone that I work with in the lab at Durham University and the collaborators at Newcastle University and the Fargo Hair Institute. Thank you.
Question? So did you, I'll ask you the same question, the heterogeneity of the spheres or whatever, between made from dermal sheath versus dermal papilla, is there a difference?
In what regard?
When you compare them to each other, to
the sizes and
gene expression.
So we've not looked at the gene expression, or I've not looked at the gene expression between dermal papilla and dermal sheath. I'm actually giving another talk tomorrow where I'm trying to restore DP character to the dermal sheath. So there are big differences between DP spheres retain hair follicle inductivity, whereas the sheath ones don't. But specifically at a gene level, I couldn't tell you.
We'd like to thank all the speakers, and thank you all for staying until the end. Thanks.